This program made possible in part by grants from the UCF College of Health and Public Affairs, which promotes excellence in undergraduate and graduate education, research, and community service in health-related professions and public affairs. And the UCF College of Medicine, where our goal is to become America's leading partnership medical school and a national leader in medical education and research. And the UCF College of Nursing, which offers high-quality, innovative academic programs that reflect the healthcare needs of a changing population and meet the needs of today's students.
Hello everyone, welcome to For Your Health. I'm Ed Heiland.
And I'm Charna Wiese. On today's show, lasers have helped to revolutionize some areas of medicine like dermatology and ophthalmology. Now the cutting-edge use of light is poised to change cancer treatment as we know it.
I'm Alicia Callanan Mandigo. Compelling new research is changing our understanding of cancer and DNA. I'll have that story.
And speech pathology is an extremely varied field that's increasing in popularity. That's coming up. But first, an update on health news from UCF.
Michael Frumkin, Dean of the School of Social Work and Human Services at Eastern Washington University, is the new Dean of UCF's College of Health and Public Affairs. Under his direction in the last 10 years at Eastern Washington, the social work program more than tripled in size and became a standalone school. He starts at UCF July 1st. UCF researchers have found a new way by measuring how many gold nanoparticles bind to a bacteria cell to speed identifying bacterial infections in patients so proper treatment can start more quickly. Not only is the new technique fast, but it's very sensitive, and it works in complex body fluids like blood.
Most assays, they have a detection limit. So if you have one bacteria or one or two, it's very difficult to detect them. But this one is totally the opposite. The lower number of bacteria you have, the more of a signal you will have. And as I said, you can do it in this turbid solution. Oftentimes, people report these femtomolar and attomolar detection levels, but they only do it in water or PBS, in very clear, pristine solutions. But in here, we're doing it in very dirty solutions.
Researchers say they can use existing equipment to analyze up to 96 samples at a time, speeding the process even more. This is Jerry Klein for Your Health.
Researchers are delving much deeper into the mysteries of DNA, and they're finding that it plays a role in things they hadn't previously suspected, like when and why a tumor develops. Alicia Callanan Mandigo has the story.
This lab within UCF's Department of Biomedical Sciences is breaking some of the mysteries of DNA.
We're trying to understand, mechanisms of gene control as it pertains to regulation of cell growth.
Every gene we have has a duplicate, as Dr. Muller illustrates here. If you're born with one strand damaged, you have a backup. But things like smoking could damage the backup strand.
And this, by the way, could take 20 years or more. And when that happens, you now are mutated in both copies of the gene. If that gene is important in maintaining a negative growth status in whatever tissue it's in, then this will override, and you'll get growth promotion and you'll get cancer.
Here's a kind of simple way to think about what genes are doing in our bodies. You have two kinds of DNA. One kind is an accelerator. It accelerates growth, like the accelerator in your car. The other kind stops growth, like the brake. One keeps the other in check. It's a critical factor in Muller's main research vehicle, which looks at gene silencing.
This is a relatively new area, like in the last maybe 15 years. It's been discovered that genes get silenced, and you don't always have to make a mutation in a tumor suppressor gene to kill it. sometimes the genes get turned off like a light switch. So if you silence, I call it inappropriate silencing. If you silence a gene by mistake, that's again getting back to our accelerator and brake model. If you silence the brake, then the accelerator takes over, and you have the same thing.
His work is part of an international collaboration. When researchers in Italy and Colombia came across a suspected incident of gene silencing, they asked Muller's UCF team to help.
And we devised the experiments to go in and map the action of the silencing equipment on that gene that they were looking at. And we showed that, in fact, it was being silenced by the maintenance machinery.
It's fascinating research that hopefully one day will lead to better cancer treatments and even earlier detection.
And one thing I really like about UCF and the environment here is the administration is favorably disposed to promoting a research environment.
I'm Alicia Callanan Mandigo for your health.
Finding the key to these DNA repair mechanisms could perhaps lead to better cancer treatments.
Lasers, always exciting to talk about, may also lead the way to better cancer treatments. The science behind it is called photonics, and photonics could affect your medical care in two exciting ways.
Joining us now is Dr. Kevin Bellfield. He's the department chair and professor of organic and polymer chemistry. We have so much to talk about, and first is so much to define for us and so many of the people watching. People don't usually associate chemistry and lasers. It's really two modalities, and I would bet, I'd bet that you didn't think you would be working with lasers when you started out to be a chemist.
That's correct. That's correct. But it's a fairly old field, but it's gone through a new emergence with the advent of lasers called photochemistry, in which one uses light to drive chemistry, and so that's kind of the marriage that we're seeing in the kind of work that I do and others do, where we use lasers to drive chemical reactions.
Yeah, well, those chemical reactions, as we alluded to in the lead-in, can help lead to medical treatments. I know from previous discussions you've been working with a fascinating area where you're actually utilizing a laser to trigger a reaction that can help fight cancer. You want to get us into that a little bit more? Sure,
sure. Well, in general, in photonics, that's a technology. It's a technology of light, basically, or applications of light. Formally, it's a science and engineering of generating, harnessing, and detecting photons, or radiant energy, and that energy can range anywhere from the X-ray that we see in the use of X-rays for diagnosis, all the way out through the ultraviolet light, the visible region, the infrared, and then going out into the microwave region, where we use microwaves quite commonly in our microwave ovens and in telecommunications, out into the radio frequency range. And so there's a vast spectrum of energies that we can harness to do things in chemistry. And the laser, after that was invented in around 1960, really has opened up a lot of possibilities to both study some fundamental work in chemistry and then develop applications of the chemistry. We've been using laser radiation in a couple of very interesting ways. One is in using it in advanced microscopy to interrogate cells and to look at where probes go into different regions of cells, and to help cell biologists determine mechanisms of action in cells. So we've been developing materials that will interact with very specific types of laser radiation for this type of imaging. And one of the areas is developing agents that can be used to detect tumors at very early stages, before they may be detectable by X-ray or by other means. And that's involved using or modifying these fluorescent probes, modifying the chemical structure so they will have some selectivity to target just tumor cells and not healthy cells. And so we started out that work here at UCF about 7 or 8 years ago. And sort of somewhere along the path, we recognized that we've been using light to drive chemical reactions, and there's a whole area of light-activated medicine called photodynamic therapy, and it's used to treat some types of cancer. And so we thought that, well, we're developing these structures that will target tumor cells to allow us to visualize the tumor cells by irradiating them with light and then looking at their fluorescence. Why can't we change the molecular structure a little bit, so instead of now detecting the tumor, if we already have one agent that we can send in to detect where the tumor is, we can then follow that with another agent that has the same chemical selectivity for the tumor cell, but now we shine a different wavelength of light on it, and it either generates reactive species that will then kill the tumor cells or releases a drug.
It's like a light-triggered chemotherapy of sorts.
Exactly.
So rather than putting the chemotherapy in the whole body, which affects the whole body, it would only be triggered by the light, the agent, and the light would be where the tumor was?
That's correct. So the agent itself has some selectivity to tumor cells, and so should localize in higher concentrations in tumor cells. But the agent itself is not toxic. It doesn't generate a toxic species until it interacts with light. And so one can shine light on just where the tumor is localized, and then localize the formation of the cytotoxic, or in essence the anti-cancer agent. And that greatly minimizes side effects. And so one cuts down on destruction of healthy cells. And so the side effects should be very minimal. And in fact, there are some FDA-approved uses of photodynamic therapy now to treat certain types of melanoma, certain types of esophageal cancer. And those are typically surface-type cancers where one can get the light directly to the surface.
So right at this point, you're looking at things that are easy to get to. Is it going beyond that to places, excuse the expression, where the light don't shine?
Right. That's a good analogy. In fact, the field has gotten to the point where one can get light to surfaces by different ways. Endoscopy is one method of delivering a fiber optic light or an optical fiber to where one wants, and then irradiating, say, a tumor inside the stomach or even into the lungs. The technology that we've been working in for about a decade now is called two-photon absorption. And this two-photon absorption is a very, very interesting process. It's a process that myself and most photochemists were probably unaware of more than a decade ago, and now it's becoming a staple in modern photochemistry. But it uses wavelengths of light that normally would transmit straight through material. Material would not absorb the light. But with the use of very special lasers, high-power lasers, and these special wavelengths out into the near infrared, just beyond where we can see, we can focus the beam and get the energy very high to allow excitation of materials that normally would just transmit the light. And what this does is it allows us to use this near infrared light that will transmit through biological material. In fact, will transmit through your skin and through tissue, and without causing any damage, without any absorption. So that means nothing will happen. But one can focus the light, and then in the area where it's focused, for example, where the tumor is, the energy is high enough to cause this two-photon excitation. So the energy of these two long wavelength photons basically combine and cause the same kind of photochemistry you would get if you used ultraviolet or visible light. But you have the advantage now that the ultraviolet and visible light will simply be absorbed on the surface and not allow you to penetrate to deep tumors. This near-infrared light will allow you to penetrate right down through the skin, through tissue, and then localize millimeters or centimeters deep into tissue, and only allow the excitation to occur where you focus the beam.
Wow.
That's a fascinating area. We've got much more to talk about. We're going to have to take a short break here, but we'll be right back talking with Kevin Bellfield, head of the chemistry department at UCF. And we're also going to give you an update on our new medical school, so don't go away.
We're back. We're talking with Dr. Kevin Bellfield about these amazing things that lasers are going to be able to do with us. And unfortunately, so many of us in the media, we talk about things that are in research, in development, and then people are watching and they're wanting to know, when is this going to be available? Sometimes it won't be for 10 years, sometimes it'll be 5. You said that the lasers are being used in some cases for skin cancers, melanoma, esophageal. How about the new one, the Photon 2 you're working on? How far in the future?
The two-photon technology is still in the research stage, but since the medical community is embracing the conventional photodynamic therapy more and more, and they're embracing the use of lasers, that we would anticipate probably within 5 years, I would say, that we'll see at least some clinical studies that will be done. Already, macular degeneration is age-related macular degeneration is a large cause of blindness in the elderly. There's lasers now that have replaced surgical techniques and other techniques to go in and stop plug the leaky arteries that are leaking the blood into the eye and causing problems. So there's more and more acceptance of lasers in the medical community. And as medicine, as the administration of drugs has been widely accepted, or chemotherapeutics by the medical community, now with more laser use in medicine, we think there'll be more acceptance, especially when one sees the benefits of the reduced side effects. If you've known anybody that's gone through chemotherapy, the side effects are pretty devastating, and oftentimes with serious cancers, the patient dies of side effects of the chemotherapy drug before they die of the outcome of the cancer itself. And in this way, if one can reduce or practically eliminate side effects, and one can concentrate just on actually stopping the cancer progression.
And all this has a domino effect, does it not? I mean, just we were discussing in the beginning of the program about how you started getting into lasers and realizing how lasers can work very closely with some of the chemical reactions that you were seeking. You're starting to see work where you do this and then somebody else picks up on a part of your work, and you look back at what somebody else is doing, and the community begins to somewhat be feeding off of each other, if you will, in terms of trying to build on this, and you incorporate their ideas and then move on to the next step, and then hopefully we'll make the whole process move a little faster.
That's right. That's right. And yeah, there's a lot of work that we that we look at in the conventional drug delivery field, where people are developing drugs to target different types of cells or to target different organelles within cells. We also look at that and incorporate that into our designs. So there's a lot to learn from people working in other fields.
We haven't. Oh, I'm sorry. I thought you were done. Go ahead.
But what this is really doing is setting the stage for a relatively new branch of medicine called photomedicine, and so I think we'll be hearing a lot more about that in the future.
We haven't talked about polymers yet, and that's it's almost science fiction to me, thinking about the possibility of plastics that can move and stretch and maybe even work like a muscle.
Or maybe store things inside, such as energy and images, as I know you're working with your 3D optics.
Right. Yeah. This two-photon absorption process that allows us to move a beam around and excite very specifically wherever we want to, or in 3 dimensions. We use the exact same principles that we're using in the photomedicine or the tumor imaging for 3-dimensional optical data storage to develop very high data capacity storage media. So, for example, in the middle 80s, the CD was introduced, and so that replaced the floppy disk. And some of us probably remember floppy disks. And of course, our students may not remember those. But the CD was revolutionary. You know, it gave us about 750 megabytes of storage on this disk. And about 10 years later, we came out with a DVD, and now we can have movies on DVD. Before the CD was, we could have music on the CDs, but it didn't have enough capacity to store video images. Or large video images. The DVD gave us that capacity, but to go further and to store even higher resolution video images or a lot more data, one desires even greater storage density. So the DVD allowed greater storage capacity than the CD, and that was accomplished again through photonics by going to shorter wavelengths of light and going to lenses that have what's called a higher numerical aperture. The higher the numerical aperture, it means that the smaller the focus can be, the volume of the focus. So basically by using shorter wavelength of light and focusing into a smaller spot, one got higher data density. Well, just last year, the Blu-ray technology was introduced, and so that increased from the DVD storage density of or capacity of about 2.5 gigabytes. It increased by an order of magnitude to 25 gigabytes. But now, to be of real practical use, to help physicians, for example, download high-resolution X-ray images, MRI images, CAT scan data, all for a patient, and to have available at the point of treatment requires enormous capacity for storage density, and typically requires getting files from one specialist's office and another specialist's office, and hopefully they'll arrive on time when the patient is there. But to be able to transfer all that data over the internet and then download it and put it in a high-capacity storage medium in short order would be a huge advantage for the medical community. So the technology that we're developing is designed to now go beyond what the storage capacity is of the Blu-ray disc. And the Blu-ray disc was, again, an evolution. They went to shorter wavelength yet again, higher numerical aperture. But basically, they've reached, the technology has reached the capacity of increasing the storage density by going that route, by now taking advantage of the three-dimensional storage that we're able to do and go and move away from just 1 or 2 layers of data storage, but being able to store perhaps 100 to 500 layers of information. So now we can realize, 1, 2, 3 orders of magnitude, perhaps more data capacity per disk than the current technology.
Gigabytes and gigabytes of information, all stored on polymers there. Terabytes.
That's right.
Add it all up.
Yeah.
Kevin Bellfield, thank you so much for coming by and joining us once again.
Great, thank you.
Well, there's lots of changes and new plans for the UCF College of Medicine coming to our Lake Nona campus in 2009. Here's a look.
University of Central Florida's College of Medicine received preliminary accreditation in February. The accrediting agency said that the plan for the college is worthy of emulation in several areas. For example, it does a good job of using university resources such as information technology.
They said that our College of Medicine had the potential to be a national model, once again, for other medical schools in our knowledge management and assessment systems. And these are currently in development. And I just want you to know how pleased I am that we were able to achieve this in our very first site visit.
President John Hitt added to the list of university resources that will help make the college a model for 21st century medical education.
Bringing in the tremendous capabilities we have in simulation and training into the curriculum and the tremendous advantages in imaging that our Center for Optics and Photonics, Center of Excellence in Optics and Photonics and College of Optics and Photonics make possible here at the university. So we've got some huge advantages in Orlando and specifically at UCF.
The college expects to begin reviewing student applications in summer 2008 and open its doors in fall 2009.
Speech pathology is a career that takes its practitioners to a lot of different settings, as Alicia Callanan Mendigo reports.
So who can tell me, where did you go on your trip last week?
This is a second career for speech pathologist Angie Greenwood. She used to be a middle school band director, but decided she wanted to work more closely with her students. So she went back to school at UCF and became a speech pathologist.
And I like this field because it gives me an opportunity to do lots of different things. I could work in lots of different settings. I could have a variety of focuses like the speech or the language, swallowing, rehab in hospitals. There's a lot of opportunities.
A lot of us think of school speech pathologists as working predominantly with elementary age students, but there are lots of high school students who work with them too.
A lot of them are regular students on a general diploma track. They go to science and math and reading like everybody else. And typically with those students, I might pull them out of class once a week, do some activities with them, some reading and writing sorts of things to help boost their language skills in the classroom. and then they would go back to class. I also have some kids that are on special diploma and work with them kind of in the same sort of areas, reading and writing, and a lot of it is reading and writing academically based.
These kids are disabled, and Greenwood is working with them on associating words with specific objects, but they're only one example of the kids a speech pathologist might work with.
I like the public schools because you see a variety of different issues. If you were in a private practice, you may have a practice that focuses on voice disorders, or you may have a practice that focuses on articulation or language, but in a public school, you're going to see everything.
And that's part of what drew her to speech pathology. It provided a challenging new career that allowed her to continue working in a school environment.
I love this job because I can make a difference. I feel like each time I sit down and I interact with a child, I can have make an impact personally. Some of these kids, they don't have adults in their lives that care about them, that listen to them, that encourage them to communicate. And so I can make a personal impact, I can make an academic impact. If I can help someone to communicate in a way that they haven't been able to before, then that's rewarding. I mean, that's life-changing for those folks, and it really is fun to watch that happen every day.
Eat. You got it.
I'm Alicia Callanan Mandigo for Your Health.
And that's it for us today. I'm Charna Davis Wiese.
And I'm Ed Heiland. Thanks for joining us, and we will see you next time on For Your Health.